Okay, hi everybody. I'd like to thank the organizers for arranging this session and for inviting me to participate. I've seen what an incredible amount of work this is, and I think we should all be very grateful to them for their efforts. Okay, so oxytocin is a 9-amino acid peptide that is known to be involved in modulating social behavior across a wide range of vertebrate species. Oxytocin is synthesized in the supraoptic and paraventricular nuclei of the hypothalamus, and it's secreted by way of the posterior pituitary into the peripheral circulation, but it's also secreted from the hypothalamus up into the brain, where it can have effects on behavior. And the main experimental tool that we have available to manipulate oxytocin levels in the human brain is intranasal oxytocin administration, which we now know can penetrate the blood-brain barrier and can access the brain. So our lab has been investigating the question, how does intranasal oxytocin modulate the brain's response to positive and negative social interactions in both men and women? And so we image our participants' brain function with fMRI as they perform a dyadic social interaction task with other people who are outside the scanner. And the task that we use is called the Iterated Prisoner's Dilemma game. And this is, I'm sure this is a game many of you are familiar with, but this is a very simple two-player game in which you and another person choose to either cooperate or defect, and you receive a payoff that depends on the interaction of your two choices. So if you cooperate and your partner reciprocates that cooperation, you both receive $2, and we consider that a positive social interaction. On the other hand, if you cooperate and your partner fails to reciprocate, then they benefit at your expense, and we consider that a negative social interaction. And so we're interested in how the brain responds to these positive and negative social interactions and how those neural responses are modulated by intranasal oxytocin. So we conducted a placebo-controlled double-blind study in which approximately 300 participants were randomized to treatment with either 24 units of intranasal oxytocin, 20 units of intranasal vasopressin, or a placebo, and about half of all participants were female. And this just shows you what a single trial of the task looks like. So we show the subject the round number. Then player one, or the first mover in the game, chooses to cooperate or defect. That choice is immediately revealed to player two, and then player two then chooses, and the outcome is revealed to both players. And we're particularly interested in what the brain is doing during this epoch when the outcome is displayed to both participants. So today I'm just going to talk about the session in which the participant is playing in the role of player one, or first mover in the game, against a same-sex human partner, and they play 30 consecutive rounds of the game. They're led to believe they're playing with a same-sex human partner. They meet this person who's a human confederate, shake hands with them, but in actuality, the choices are administered by a pre-programmed computer algorithm, and that algorithm is responsive to the choices that the subject makes. And specifically, it reciprocates defection 90% of the time, and it reciprocates cooperation 2/3 of the time. So I'm going to tell you about 3 different sets of results today. First, the effects of oxytocin on the neural response to positive social interactions. Then the effects of oxytocin on the neural response to negative social interactions. And finally, the effects of oxytocin on functional connectivity during social interactions. So here we're just looking at activation at baseline in the placebo group. So what you see are areas of the brain that are more active in response to reciprocated cooperation, which is the positive social interaction, compared to unreciprocated cooperation, which is the negative social interaction. And you can see that in both men and women, there's activation in the medial orbital frontal cortex, which among other things is involved in reward processing. But we see more activation in women than we do in men. And additionally, in women, we see this bilateral activation in the nucleus accumbens that we don't see in men. So one possible interpretation of this result is that at baseline in the placebo group, women find same-sex positive social interactions to be either more rewarding or more salient than men do. So then we can ask, well, how does oxytocin modulate this pattern of brain activity? And here you can see the results in men. So this is showing you that oxytocin increases the response to reciprocated cooperation in men in the regions that you see here, including the nucleus accumbens. So this might imply that oxytocin renders these positive social interactions more rewarding or more salient in men. On the other hand, in women, we find that oxytocin decreases the neural response to positive social interactions. And if we look at the direct contrast of oxytocin effects in men and in women, and this is now thresholded according to, you can't see that, but according to standard recommendations for adequately protecting against false positives. So P of less than 0.001 at the voxel level and P of less than 0.05 at the cluster level. So here in the caudate and then putamen, we see that in the placebo group, women have a stronger response than men do. But if we give men oxytocin, we boost them up to the level of women in the placebo group. And then sort of unexpectedly, if we give women oxytocin, rather than boosting them up further, we decrease them back down to the level of men in the placebo group. So this was unexpected to us, but if you look through the literature, there are some parallel findings in non-human experimental animal studies. So this is a study done in hamsters where on the y-axis you have a measure of social reward, and you see the same sort of pattern where in the saline group, the females have a stronger preference for this social chamber than males do. But if you give males oxytocin, you can boost them up to the level of females in the placebo group. And if you give females oxytocin, you decrease them down to the level of males in the placebo group. So sort of post hoc, we had to try to come up with an explanation for these findings. And we thought, well, maybe there's a nonlinear inverted U-shaped dose-response function. And so here we have levels of oxytocin in the brain on the x-axis and the neural response on the y-axis. And we're suggesting that at baseline, men might start off, men might start off here further to the left, whereas women may start off closer to the peak of the dose-response function. So there is one paper showing that women have higher levels of oxytocin in their cerebral spinal fluid than men do. And so our suggestion is that when we give men 24 units of intranasal oxytocin, we push them up to the level of women at baseline, but when we give women more oxytocin, we kind of push them over the edge back down to the level of men. Okay, and then this is just a really brief digression, but more recently we've been doing a lot of research with men who are fathers, human fathers, and when we show them pictures of their toddlers, we also find that intranasal oxytocin increases their brain's response in the caudate nucleus and the dorsal anterior cingulate cortex. So I thought it was just interesting that multiple different types of kind of positive social stimuli, oxytocin seems to enhance the neural response to those in men. We've also identified a SNP in the gene that codes for the oxytocin receptor that modulates these effects. So this is RS53576, which is in the third intron of the oxytocin receptor gene. And we've been able to show that the results that I just showed you are really driven by a particular genotype at that SNP. So individuals who have the GG genotype here have the same pattern I was just talking about, whereas the A allele carriers don't have that pattern. Okay, and then I'm going to skip this. But I wanted to go on and talk about, well, how does oxytocin modulate the response to unreciprocated cooperation? So that's the negative social interaction. And here you can see, again, just at baseline, these are areas that are more responsive to unreciprocated compared to reciprocated cooperation. And so those include both the amygdala and the anterior insula, which have many different functions, but one of their functions is clearly they're involved in stress and anxiety. And so it's interesting that in men, intranasal oxytocin attenuates that response. So it decreases their response to unreciprocated cooperation in both the insula and the amygdala. And that raises the possibility that that oxytocin is rendering these negative social interactions less stressful or less anxiety-provoking. And then once again, we see sex differences. So in women, this time we find no effect of oxytocin on the response to unreciprocated cooperation. And then just again, these were unexpected results, but if we look at the literature, I thought this was an interesting convergence. This is a study where they measured how much distress participants reported in response to the Trier social stress test. And this is just showing that in men, oxytocin significantly decreased the amount of stress that they reported, whereas it had no effect in women. So that kind of parallels the results that I was just showing. Okay, and then the last result I wanted to show was how oxytocin modulates functional connectivity. And here what we did is we specified what we call a social behavioral neural network, which is loosely based on an analogous one that's been defined in rodents. But basically we define regions of interest bilaterally in each of the regions that you see shown here. So these are the nucleus accumbens, the amygdala, this is the nucleus basalis, the anterior insula, the orbital frontal cortex, the lateral septum, and the ventral tegmental area. And so there's a total of 182 possible connections among all of these regions. And we just asked in a PPI analysis, a psychophysiological interaction analysis, whether oxytocin modulated functional connectivity between any of those two nodes. And so specifically during cooperative social interactions, so in response to the outcome of mutual cooperation. And so here, first of all, is the result for women, where you see that there's a small number of connections that where oxytocin increases functional connectivity and a small number where oxytocin decreases functional connectivity. But then when we look at men, it's a pretty stark difference where there's a large number of connections where functional connectivity is increased by oxytocin. And the two nodes of the network with the largest number of modulated connections are the nucleus basalis and the lateral septum. And these areas are of interest because we know that they're two areas that have a very high density of oxytocin receptors in the human brain. So it makes sense for us to when we administer exogenous oxytocin, it would make sense for us to see changes in brain activity in areas that have a lot of receptors to sense that oxytocin. And so we think oxytocin may be working through these areas to kind of synchronize that network and thereby enhance the salience of these positive social interactions. So So that was my third point. And then, so I guess in conclusion, there's a lot of interest in among psychiatrists in the potential use of oxytocin to treat social behavioral disorders. But our findings suggest the need for some caution in doing this. And so first of all, the effects differ for men and women. So, you can imagine a lot of people have talked about there being deficits in social motivation and social reward in autism. Our data would suggest that using oxytocin in men who have autism is probably a reasonable strategy, but using it in women might not be, and it might actually be harmful. So we need to think about that. Also, with very few exceptions, there haven't been many dose-response studies. So all I did today was talk about 24 units of oxytocin, but maybe if we gave 8 units, the response profile would look very different in women than what we see. Also, effects may differ between healthy subjects and patients. So today I showed you data from healthy patients, from healthy subjects, but other people are looking at patients. And it could be that the effects of endogenous and exogenous oxytocin differ. So we're kind of spraying oxytocin everywhere in the brain, but if you had a drug that would release oxytocin from neurons that have it in a more targeted way, you might get different results. And there could be differences between what you get when you give a single dose of oxytocin versus if you have repeated long-term administration and chronic dosing of oxytocin. So all of these are things that we need to address in the future. And finally, I'd like to thank all of the individuals listed here who've contributed in various ways to this work, and thank you for your attention.